Okay, good morning. Let's get started. So today we're going to finish all the lipoprotein metabolism, the session. And for the Tuesday, next Tuesday, we are going to talk about essential fatty acids. I think we probably have about 1 hour time left, so I'm going to do the review session, right? Like last time, you guys can just ask me questions based on whatever study guide or something that you are not very clear. And next question. Oh, about the next Tuesday, right? We have exam 3, right, next Thursday. So next Tuesday, we need to finish one more session, like essential fatty acids. And I think we probably just need 1 hour for that session. So we about an hour, left for review session for the next Thursday exam, right? And for the coverage for next Thursday, I think all the lipid metabolism, right? And do you guys think you will need some extra time in addition to next Tuesday? You need a review session? Do I need to, make like 1 hour or so at different time? I mean, not like a class time. Need it? Okay, yeah, I'll try to schedule a review session, right? Maybe sometime, let's see, Wednesday, and maybe sometime next Wednesday. All right, so fatty liver. So we already talked a little bit about fatty liver disease last class. So we know that the main cause of fatty liver disease is due to the imbalance between triglyceride formation and triglyceride export. For example, if we have either too much triglyceride being synthesized, or if we cannot export triglyceride out of the liver, then we're causing a accumulation of triglyceride in the liver and fatty liver disease. Okay, and we also talk about the possible defect that can cause the fatty liver disease, right? And two types of reasons that causing the so-called non-alcoholic fatty liver disease, right? So fatty liver disease in general has two different types. One is non-alcoholic, and the other one alcoholic, right? Means that caused by alcoholism or non-alcoholism factors. So the two main types for non-alcoholic fatty liver disease. First type is causing too much fatty acids that being circulating in the plasma, right? And this kind of situation, we know that, for example, type 2 diabetes, particularly type 2 diabetes, and we have a lot of fatty acids being released from adipose tissue and circulating in the plasma. And all the other reason, it could be the chronic intake of a high-fat diet. We eat high-fat diet all the time. Then these are the two important reasons for increasing plasma fatty acid levels, and liver picks fatty acids and synthesize triglyceride, right? So then we have too much triglyceride being synthesized and in the liver and causing liver disease. And the other reason is that the metabolic block of the production of lipoproteins. We know that lipoprotein is needed to export triglyceride right out of the liver. So if for some reason that we cannot make enough lipoproteins, particularly VLDL, in the liver, then we cannot, efficiently output the triglycerides. And so there could be multiple reasons. For example, somehow the apoprotein cannot be synthesized for some most likely genetic mutation or genetic reason. So then you cannot make functional lipoproteins, right? Particularly VLDL, or VLDL or the lipoprotein cannot be secreted. And the other reason, for example, the lack of the phospholipids, which is needed for the surface of a lipoproteins, right? For all this reason that we cannot make functional lipoproteins of VLDL, particularly. So then we cannot, transport triglyceride out of the liver, so leading to the accumulation of triglyceride in the liver. And alcoholism can also cause a fatty liver disease, which is very common, right? And first of all, we wanted to just describe a little bit about how alcohol metabolized in the body, particularly in the liver. So so alcoholism basically can ethanol, right, can be oxidized to acetaldehyde, and by this enzyme called alcohol dehydrogenase in the liver, and generating a lot of NADH. And this NADH produced from alcohol metabolism, right, ethanol metabolism, and can compete with the normal pathway, the NADH that produced by the normal pathways, right, which for the oxidative phosphorylation. So that will so too much NADH that produced from ethanol metabolism can inhibit the fatty acid oxidation and oxidative phosphorylation, right? So once the fatty acids being oxidation being inhibits oxidation. So then you have a lot of fatty acids being cannot be oxidized efficiently, right? So then being used for synthesis of triglyceride in the liver. So this is the main reason, for why alcoholism can cause the fatty liver disease. Basically, it's too much triglyceride being synthesized from fatty acids, which cannot be efficiently oxidized because of this ethanol metabolism, right, leading to increased levels of NADH and inhibits fatty acid oxidation. Now let's switch to the cholesterol metabolism or homeostasis. Actually, two different aspects. One, we know that cholesterol can present in the plasma, and the cholesterol is also present in the cells, right? So the two different type of homeostasis. Plasma homeostasis means that levels of cholesterol need to be maintained at the normal range, normal levels, and also inside of cells, cholesterol also need to be maintained in the normal range. We need cholesterol for a number of reasons, right? But we don't need too much cholesterol. So first of all, in the plasma, cholesterol normally present in the plasma in the LDL, right? So we usually call LDL cholesterol. So LDL cholesterol level is very important, and the levels of LDL cholesterol usually is controlled or regulated by very important factors that we talked about earlier. Remember the LDL receptors, right? The uptake of LDL is a key factor that control the levels of LDL cholesterol in the circulation. So, for example, if LDL receptor somehow is deficient or cannot function well, then the LDL particle cannot be picked up, right, by the cells, so then the remaining in the circulation can increase the levels of LDL cholesterol in the circulation. So try to remember the LDL receptor plays a key role in controlling the plasma levels of LDL cholesterol, right? And I just wanted to ask you again, which tissues that picks up most of LDL? Liver, right? So because of the liver express very high levels of LDL receptors, right? So liver is the main tissue that picks up the LDL. Okay, now let's talk about intracellular cholesterol homeostasis. Mean that we only need just right amount of cholesterol in the cells, and we don't need that much, right? And we already talked about the HDL cycles, remember? So HDL cycles also involved in the regulation of intracellular cholesterol homeostasis, cholesterol levels, right? So this just represents a cell. I just wanted to summarize the pathway that regulates the cholesterol level. So in the cells, there is so-called free cholesterol pools, right? So free cholesterol basically present in which proportion of the cells? Do you guys know? Membranes, right? Usually free cholesterol present in membrane, cellular membrane. You know, inside of cell there are a lot of different organelles, and organelles as the membranes. So they present the membrane, either cell plasma membrane or the intracellular membranes, right? So this intracellular free cholesterol pool can be maintained by two major pathways. One is the you're adding cholesterol to the pool, and the other pathway is that you remove the cholesterol from the pool, right? So for adding free cholesterol to the pool, there are actually three different pathways. So first pathway is that pickup, right? Cell can pick up cholesterol from the circulation, right, through the LDL receptor mediated mechanism. So, for example, that the LDL receptor plays a major role that mediates the pickup of LDL cholesterol, right, into the cells, so contribute to the free cholesterol pool. And there's three different, ways, right? The one is the LDL receptor mediated pathways, and the second pathway is the scavenger receptors, SRB1 receptors, and it can also mediate the uptake of LDL. And this pathway is called a non-regulated pathway, right? It's a little bit different from LDL receptor mediated pathways. And the third pathway is that some of the cholesterol can also directly deposit on the surface of plasma membrane, right? So this free cholesterol C represents the cholesterol, free cholesterol. This cholesterol can also go to the free cholesterol pool, contribute to the free cholesterol pool. But the LDL receptor is the major pathways, right? Compared to the other two, scavenger receptor and the free cholesterol pathway. And the second pathway is the synthesis, right? We know that any type of cells are able to synthesize cholesterol. So cholesterol synthesis is also contributed to adding free cholesterol to the pool, right? Second pathway. And the third pathway is the hydrolysis of cholesterol esters. So cholesterol present in the two forms in the cells, the free cholesterol and cholesterol esters. So if somehow hydrolysis of cholesterol esters increase, then you can release more free cholesterol, right? So the three pathways that I highlight here is adding free cholesterol to the pool. That means if any of these three pathways activities increase, will leading to increase free cholesterol in the cells, right? And the other three pathways that can remove the free cholesterol in the cells, out of the cells, right? So first pathway, which is most important pathway, we already talked about it, right? It's related to the cholesterol, HDL cycles, reverse cholesterol transport pathway, right? So, which is, ABCA1 receptor that's located on the cell surface and can transport free cholesterol to the pre-beta HDL, right? Remember this pathway. So this is the most important pathway that can remove free cholesterol out of the cells. And the second is that some of the cholesterol can be utilized for the synthesis of sterols in the cells, right? So this pathway can also get rid of some of the free cholesterols. And the third pathway is that free cholesterol can also be converted to cholesterol esters, right? So three pathways that contribute to the removal of cholesterol from free cholesterol pool in the cells. So all these pathways that need to work together and to balance the cholesterol levels in the cells. We only need a certain amount, right? And if we have too much, and then this three pathway probably will be more active, try to remove extra triglyceride out of the pool, out of the cells, right? So question here, you probably would easier to understand now. For example, what kind of factors that can causing cellular cholesterol levels increase, right? First, increased uptake or increased synthesis, right? Or increased hydrolysis of a cholesterol esters, right? Basically increase this three pathways that adding cholesterol to the free cholesterol to the pool. Or you can have reduced the disposal pathways, for example, reduced this efflux of cholesterol by HDL recycles, right? Or you have somehow decreased the utilization of cholesterol pathways, or decrease the acidification of free cholesterol to cholesterol esters. For example, if ACAT enzyme activity is decreased, that we cannot efficiently esterify free cholesterol to cholesterol esters, right? If we reduce these pathways, right? So either increase this pathway or reduce this disposal pathway, and can all contribute to the increase intracellular cholesterol levels, which is not good, right? Which causing the atherosclerosis if this happening in the macrophage in artery wall. Okay, so here the bottom box actually try to talk about how cellular cholesterol levels regulate the LDL uptake and the cholesterol synthesis, right? Remember, so if we have too much free cholesterol inside of cells, and we have a sort of cellular self-regulating mechanism, right? So we wanted to slow down, uptake from the circulation. So increased free cholesterol inside of cells can inhibit the LDL receptor synthesis, right? So decrease LDL receptor on the cell surface, so that can decrease uptake of LDL, right, from the circulation into the cell. And the second thing is we already learned from the cholesterol synthesis pathway, right? Cholesterol levels itself can also regulate the cholesterol synthesis pathway. So increased free cholesterol levels can inhibit the cholesterol synthesis by one of the important transcription factor. Do you guys still remember? which transcription factor that can be inhibited by cholesterol levels? SIBP, yes. So this is the pathway, right? So inhibits, the cholesterol synthesis. And lastly, the ATCA, which is enzyme catalyzes acidification of free cholesterol to cholesterol esters, right? So increased free cholesterol can stimulate ACAT activity to allow the more free cholesterol being acidified to cholesterol esters, right? So just try to remember this, the regulatory mechanism, how the cells maintain the intracellular cholesterol homeostasis, right? So two pathways, one, the two major pathways, one the adding cholesterol to the pool, and the other major pathway is to remove cholesterol out of the cells. And we have each major pathway has the three different mechanisms, right? Okay, now any question about the cholesterol homeostasis in the circulation as well as inside of cells? Right. So cholesterol homeostasis is very important. That if anything's wrong with the cholesterol homeostasis, that will causing, the disease, particularly the atherosclerosis and cardiovascular disease. Right. It's very close related to the cardiovascular disease. So now we're going to talk about the little bit the cholesterol and relationship between cholesterol levels and coronary heart disease. So this is just a figures that showing relationship between the plasma cholesterol levels, which represent as x-axis, and 6-year coronary heart disease mortality, which the y-axis, right? So the normal range of cholesterol levels basically is less than 5, right? Millimole per liter is considered the normal range of cholesterol levels. And bottom line, if cholesterol levels are between 5 and 6.5 is considered the borderline, and cholesterol levels if higher than 6.5 and is actually not normal, right? It's the pathologic conditions, it's elevated cholesterol. And you can see that the relationship between plasma cholesterol levels and the mortality, cardiovascular disease for mortality is kind of linear relationship, right? As you've seen that the cholesterol level increase, the mortality actually increase, right? Yeah. So that means the cholesterol level is very closely associated with the or correlated with the cardiovascular disease mortality, right? So we wanted to keep our cholesterol level, blood cholesterol levels low, or at least in the normal range. And here I just wanted to show you the relationship between the saturated fatty acids in the diet and the serum levels of cholesterol. And the saturated percentage, the x-axis represents the saturated fat percentage on the total energy, right, percentage energy, and the serum cholesterol levels on the y-axis. So you can see that actually there is the positive correlation, right, between the saturated fat intake and the cholesterol levels, serum cholesterol levels. So if we take too much saturated fat, that can contribute to increase the cholesterol levels, right? So saturated fat is not, definitely, we all know, right, it's not good type of fat. We need to reduce the saturated fat in the body, in our diet. So saturated fat actually and high consumption of saturated fat contribute to the increase the circulating cholesterol levels. And polyunsaturated fatty acids actually is a good thing, right? You can see a negative correlation with the serum cholesterol levels. So if we eat more unsaturated fatty acids in the diet, and you will have less cholesterol in the circulation. So unsaturated fatty acids is a good thing. In the diet we need to have more intake more polyunsaturated fatty acids. And dietary cholesterol is also positively correlated with circulating cholesterol levels. We know that in our body's cholesterol, 50% from diet, right? So if we eat too much cholesterol, and definitely will increase the cholesterol levels. So dietary cholesterol does contribute to the circulating cholesterol levels. Okay, so now let's talk about atherosclerosis. So atherosclerosis, the major risk factor for the development of atherosclerosis is elevated plasma cholesterol, right? Again, the elevated or increased plasma cholesterol levels is a major risk factor or contributor to the development of atherosclerosis, and not triglyceride level. So triglyceride level are not clearly related to the development of atherosclerosis, right? Only high levels of cholesterol that contribute or related to the atherosclerosis. So atherosclerosis is the formation of atheroma, right? Atheroma is formed in the artery wall, right? And then block the blood flow and reduce the blood supplies to if it happens in the artery wall, then reduce the blood flow to the heart, and causing the heart attack and the other kind of serious heart disease problems, right? Cardiovascular disease. And here just give you kind of it's very clear, right? So the artery wall has the endothelium, and this is so-called the atheroma is formed right under the damaged endothelium and blocks the blood flow, right? Now let's talk about the mechanisms, how atherosclerosis is developed. In several reasons. First of all, the endothelium damage, right, for unknown reason. And this damage can cause an inflammation, right, inflammatory response in the artery wall. And so allowed a lot of inflammatory cells, for example, monocytes and macrophages, to infiltrate into the damaged artery walls, right? And this sort of initial, the process for the development of atherosclerosis or atheroma. And the second important thing is that the lipoproteins, LDL particularly, right? LDL cholesterol levels be actually deposit in this damaged site of and causing the multiplication and oxidation of LDL. So modified LDL then can be uptaken by the immune cells like the macrophages, right? And a lot of cholesterol being uptake into the macrophage and accumulate in the macrophages, right? So in other words, that LDL levels in the circulation increase is also the important thing, important factors, right? And then lastly is the formation of firm cells by macrophages. Basically, macrophages pick up a lot of oxidized LDL and cholesterol in the LDL. Right? So again, the three steps. The first of all is the damage of endothelium damage and inflammation, right, response, and attract a lot of the macrophage or monocyte to the damaged side. And then the increase the LDL levels in the circulation is a second important thing. And then the last thing is that macrophage uptake of LDL, oxidized LDL, right, and cholesterol into the cells and form the firm cells. Here I just wanted to tell you that remind you actually the three pathways that can increase uptake of LDL, right? And the first pathway or major pathway under the normal condition is LDL receptor mediated uptake of LDL cholesterol. And the second pathway is the scavenger receptor mediated the uptake of LDL. And this pathway is so-called non-regulated pathway. So as long as you have high levels of LDL, and this LDL can be taken up by the scavenger receptor pathway. And this pathway plays a very important role in the development of atherosclerosis, right? All right, so more information about atherosclerosis. So see, this represents an endothelium layer, cell layer, and lining the artery wall. And first of all, you have damage of this endothelium cell layer for some unknown reasons, and causing the inflammatory response, inflammation. You have lots of macrophages or monocyte, attract to the damaged site, and the macrophage and monocells actually eventually will differentiate or develop to the macrophages in this damaged site, right? And in the meantime, because if you have increased LDL in the circulation, right? So increased LDL in the circulation is second important thing, right? And so the LDL can be taken up by, first of all, the oxidized, and then can be picked up by the macrophages, right? And macrophages pick up a lot of LDL and cholesterol and form the firm cells. There's a lot of firm cells and forming so-called atheroma, and then block the blood flow. So three things are very important, right? For the formation of atheroma, atherosclerosis. First of all, the high levels of cholesterol, and then inflammation or damage of the artery wall, right? So all these things together contribute to the development of atherosclerosis, atheroma formation, right? Have a question?
Why does this happen with LDL but not other types of lipoproteins? Why does this happen with LDL but not HDL or IDL or VLDL?
See, HDL is a good cholesterol, right? The good lipoprotein. They remove the cholesterol, out of the cells. And LDL basically is a bad cholesterol that deposits cholesterol into the cells. And we're going to explain a little bit more detail, how actually this connected LDL levels increase and to the atherosclerosis, right? We have actually three clinical cases. So first is so-called a Tanner disease. The Tanner disease is the mutation in the ABCA1 genes. Do you guys know what is the function of the ABCA1 genes, ABCA protein? Where is ABC1 located? Yeah. Right. So it's on the surface of peripheral cells, right? For example, particularly, for example, in this case, let's talk about the macrophages, right? And then the main function of this gene protein is to transport free cholesterol from the cells and load it to pre-beta HDL, right? And it's pretty important role for the disposal of cholesterol out of cells. So what happens to the patient with this disease if the ABCA1 is not functioning? First of all, what about question here? What about the alpha HDL levels in the circulation? What do you expect? Increase or decrease? Decrease, yes. Thank you. Great. Why the decrease?
Because you can't add
To
cholesterol
pre-beta HDL, right. So you cannot forming the alpha HDL, right? So decrease or absent even in the plasma alpha HDL. What about the beta HDL? So increase, right? You know, you can convert it beta HDL to alpha HDL. So you have increased beta HDL in circulation. And what about the risk for the development of atherosclerosis? They increase, right? Because you cannot pick up, transport free cholesterol and allow HDL to pick up. So you have lots of cholesterol. It's accumulated in the macrophages, right, and forming foam cells and causing atherosclerosis, right? So this case is quite easy to understand. And then see familiar the lipoproteinemia, right? So this disease is the causing of this disease is due to the defective LDL receptors or mutations in ligand region of ApoB100. So mean the patient either they do not have or have less LDL receptor, or they have kind of mutation in ApoB100. That means that LDL cannot be efficiently picked up, right? By which tissues? Liver, right? Liver actually express most highly levels of LDL receptor, right? So liver cannot efficiently pick up the LDL from the circulation. So what happens to patient in terms of the atherosclerosis? First question is LDL level in the circulation. Well, it goes up, right? Because liver cannot efficiently pick up LDL, so LDL levels in the circulation is increased. And what about the VLDL? Why is low?
Because it doesn't have the fatty acids to pull out.
Okay, so here is the new concept here, right? We haven't talked about before. So, for example, in this patient, the LDL receptor cannot pick up LDL cholesterol from circulation. So then we're causing decreased cholesterol level inside of the liver cells, right? And that will increase the synthesis of cholesterol in the liver cells, right? And liver can synthesize VLDL, and this VLDL basically packed mostly cholesterol esters, right, and secreted into the circulation. So in other words, when liver do not have enough cholesterol and makes liver to synthesize cholesterol, and that will promote the synthesis of VLDL, which mostly contains cholesterol esters and release into the circulation. So VLDL level will also increase, but this VLDL composition of lipids is different from the normal conditions VLDL, right? It's more enriched in the cholesterol. And what about the plasma VLDL level? Cholesterol levels increase, right? We talk about this. And a risk for atherosclerosis. This is LDL receptor deficiency. So you will expect LDL receptor will not pick cholesterol from circulation into the cell. So you're probably thinking LDL cholesterol level will be decreased inside of cell, right? So in this patient, it also developed atherosclerosis. So why? Why they still develop atherosclerosis in this patient? You know, LDL receptor is not functioning and not pick up LDL cholesterol, right? It should guessing why it's still develop atherosclerosis. Yes. Right. Excellent. So because the LDL receptor deficient mostly occur in the liver, right? So liver can pick up LDL, but in the peripheral cells like macrophages, LDL pathway doesn't work, but scavenger receptor, right, still can pick up a lot of oxidized LDL because you causing the increased LDL levels, right? So the scavenger receptors that pick up lots of oxidized LDL and a lot of cholesterol be accumulate in the macrophages. Right? Right? Okay, so the last case is caused by the deficiency or mutation in ApoE, right? So ApoE, do you guys remember which lipoprotein have ApoE? Remember? Chylomicron and VLDL, right? So, so what happens to this patient? So what is function of ApoE, basically? So ApoE is important for chylomicron pickup, right? And also the IDL, remember? IDL also had the ApoE, right? So pick up by the liver. So if this patient has an ApoE deficiency, then what happens to the chylomicron remnant in the liver, in the circulation? What do you expect this chylomicron remnant, chylomicron levels in the circulation in this patient? Yes. Right. Increase because they cannot pick up by the liver, right? If this chylomicron or chylomicron remnant does not have ApoE, right, on that surface. So chylomicron and the VLDL remnant, I talk about it. Chylomicron remnant and VLDL remnant, right? Both, it will be increased. And what about ID and LDL levels? Yeah, increase, right? Because the ID and LDL basically is converted from VLDL, right? The IDL basically is the VLDL remnant, right? So both IDL and LDL levels of increase. And plasma cholesterol level is also increased, right? Because of increase the LDL and IDL levels. And same thing that the atherosclerosis will be also increased, right? You have a development of atherosclerosis, right? Any question about these cases? I guess it will probably help you to understand the whole cholesterol metabolism, right? Yes.
So here in the slide it says that LDL uptake is decreased, and it says that LDL is taken up by the V100 receptor, not the receptor. That's what you just repeated.
Yeah, but you have a more sort of, the IDL, right? You have a more VLDL. So this IDL is not mainly from the pickup, but from the precursors, right? So you have a more VLDL and IDL around, and so you can be converted to LDL, right? Okay. Any questions about these cases? Well, okay, so let's take a break and maybe we start a quiz at 11:45, right?